I'm going to spend about 40 minutes or so talking to you about work we've been doing over the last 10 years looking at arterial stiffness. So what is arterial stiffness? Well, I think we all know what we mean by that. It's how elastic or stiff our blood vessels are. And we've been assessing that for hundreds of years. This is an engraving from the 18th century showing lots of different types of pulses as described in the literature. Not sort of very objective measurements, but very descriptive, and the best the physicians at the time could do. But we've now got much better ways of assessing stiffness, and the one I'm going to really focus on to a large extent today is something called pulse wave velocity. So when your pulse travels along your blood vessels, the speed at which it goes is dependent upon how stiff that blood vessel is. So if you've got stiff blood vessels, it moves a lot more quickly. And therefore we can measure that speed of travel very easily and non-invasively with pressure probes like this applied to the carotid artery and other arteries and determine velocity. And the most common way of doing this is to measure it in the carotid and then in the femoral artery and measure what we call aortic pulse wave velocity. And for those interested in the field, this is the current gold standard measure of stiffness. So I guess that begs the question, well, why do we have elastic large arteries? Because most of us were taught that they're basically just conduits. But they're actually quite structurally interesting. They've got alternating layers of elastic tissue, which is what's shown here, and smooth muscle cells. And your aorta will have about 40 to 45 of these alternating layers. And it's this elastic tissue, and particularly the structural arrangement of that tissue, which makes the vessels elastic. So when your left ventricle ejects blood, it puts volume into the circulation. And the large arteries, and particularly the aorta, expand to take up that volume. And that has a number of very good physiological effects. It reduces your peak pressure, your systolic pressure, and it actually gives you a diastolic pressure. If we plumbed you in copper, you'd have a very, very high peak pressure, and you have no diastolic pressure. And the net effect of that is to make blood flow a lot smoother, because what your brain wants is continuous smooth blood flow. So by buffering pressure changes, that you get much more smooth blood flow. And if you're interested in energy efficiency, it makes the whole cardiovascular system much more efficient. So when your arteries get stiffer, and I'll show you some of the things driving that in a moment, of course they take up less volume. And that has some important detrimental consequences. You get a big systolic pressure and a lower diastolic pressure. That gives rise to systolic hypertension, heart failure ultimately, and in my lifetime we'll probably move from heart failure being a condition of men of my age who've survived an infarct to largely a condition of older people, particularly women, who've never had an infarct but had, quote, a touch of hypertension for many years. It also can cause myocardial ischemia and angina. You get a reduction in shear stress rate, which reduces nitric oxide production. We all believe that nitric oxide is an important antiatherogenic molecule. And finally, you get increased stress within the wall. That leads the wall to remodel, makes the vessel stiffer, and you get a vicious cycle. And it's not difficult to understand how all these things might actually increase your risk. So that's physiology. Does that translate into what you observe in practice? Well, these are very old data from Florence Haynes, published in the 1930s, who showed quite a nice relationship between stiffness here and pulse pressure, the expected relationship. And we've actually confirmed that people with systolic hypertension have stiffer blood vessels than control people. So that suggests that stiffening might be underlying the development of hypertension. And that's interesting because this form of systolic hypertension, which is shown in the red bars here, becomes increasingly predominant as you get older. And indeed, it's estimated that about 2/3 of hypertension is in this older age group. And you can see here that the predominant form there is systolic hypertension. So hypertension due to stiff arteries, nothing to do with increased resistance. Ned LaCarter recently actually showed prospectively in the Baltimore longitudinal aging study that individuals who had stiffer vessels, shown here in the red, were more likely than those who had less stiff or elastic vessels to develop hypertension during the follow-up period, providing greater evidence of causality. But I think the real reason that a lot of us are interested in arterial stiffness is because it seems to predict outcome. It's a classic slide over 10 years old now from Michel Safar's group in Paris, looking at a cohort of end-stage renal failure people. And actually, there's no sort of fiddling the statistics here. There's Kaplan-Meier plot, everyone dead, everyone alive. And what you can see is there's a very powerful discrimination in outcome between those with stiff blood vessels, a bit stiff and elastic. So you wouldn't want to be in the stiff vessel group. And that's one study, smallish cohort, end-stage renal failure. We now have lots of studies in unselected individuals, in younger individuals, showing in this meta-analysis that pulse wave velocity predicts total cardiovascular events, cardiovascular mortality, and actually seem to predict all-cause mortality in this meta-analysis. But I think, to me, the problem with this is illustrated here. Over the years, lots of people have touted new ways of assessing risk. And yet, when you add those to established risk prediction models, based largely on age, cholesterol, gender, blood pressure, glucose, they seem to add very little. And this, from the New England Journal, tried adding a range of biomarkers in and found very little change in risk prediction, suggesting that they're not really adding to what we do clinically and costs us very little. So what about pulse wave velocity? Does it really add to risk prediction? Well, we've been undertaking a meta-analysis based on individual patient data, which is what the other authors couldn't do, and looking at how adjusting for other risk factors affected the risk prediction of pulse wave velocity. And what you can see here is you go through these models, and this is the model with everything adjusted for. You get some attenuation of risk prediction, but pulse wave velocity was still actually predicting risk independently of those other variables. And another way of looking at this is to look at how many people you would change your categorization of risk. So we tend to base clinical decisions on, are you above or below, let's say, 20 or 30% 10-year risk. So if you look at this, what you find is that when you add pulse wave velocity in to the risk prediction models, you reclassify somewhere between, let's say, 14 and around 36% of individuals correctly. So there's better classification with pulse wave velocity than without using pulse wave velocity. So I hope that set the scene, and what I want to now focus on for the major part of my talk are the mechanisms driving arterial stiffness, or as it's sometimes known, arteriosclerosis. And this is an old slide from the Lancet in 1922. And it was the first slide really to suggest that pulse wave velocity increased with age. And this has been replicated in populations across the world. And for many years, the firm belief of individuals working in the area was that this was a sort of inevitable consequence of aging. And it was basically due to the fact that the elastin fibers in your aorta, and these are those from a young individual, degenerated over time and became far less elastic. Now, if you work it out, by the age of 60, you've had something like 5 billion heartbeats, assuming you've got an average heart rate. So it's not inconceivable that this is a phenomenon operating, and it's going to happen to us all. But there was an interesting observation by Albert Avolio in the 1980s, where he went to China and looked at people living in an urban environment and a rural environment, and showed that although they start off in youth with pretty similar pulse wave velocity, that over the ensuing years there was a divergence in pulse wave velocity between the two populations. And I think this is some of the strongest evidence to suggest that the age-related increase in pulse wave velocity isn't necessarily inevitable. It's not a simple consequence of getting older. It's something that we're doing to ourselves, or the environment is. And it'll be a bit like blood pressure, it'll be a complex interaction. But the fact it's not inevitable suggests that we might be able to do something about it. So these are data from Phil Hrvojenčík, published a couple of years ago. And he reviewed all the literature concerning arterial stiffness and pulse wave velocity, and looked at those studies which had found associations looking at some common risk factors such as age, blood pressure and lipid fractions. And what he found was that it was largely age and blood pressure that seemed to be predicting. Diabetes and heart rate had some effect, but the things like lipids and smoking, it was questionable about whether they were really driving arterial stiffening. And we've been fortunate to be able to look at this using some prospective data from a couple of studies. And this is from the Caerphilly Heart Study, which was set up in 1979. And as you can see here, they've been followed up over the last 20 years or so at various intervals. And therefore, we looked at the influence of baseline risk factors on pulse wave velocity measured more recently. And what we found was that if you look at the effect of cholesterol, it seemed to have no effect on pulse wave velocity. Triglycerides had a marginal effect, and smoking, again, had no effect. So this seemed to confirm what Verhofjenschik had suggested, that actually traditional cardiovascular risk factors were having little impact. And again, working with the individuals from the Whitehall 2 study, we were able to confirm these observations and show that traditional risk factors have little impact upon arterial stiffness. Now that might seem disappointing, but actually I think it's probably what you would have predicted, because these are data from a Japanese population where they actually measured their pulse wave velocity of individuals in life, and once they died they looked at atherosclerotic plaque in the aorta using Allredo staining. And they found, all right, a correlation between the two. But when you look at the magnitude of this correlation, it's actually very small. Therefore, it's not surprising that we have risk factors, we think of cardiovascular risk factors like smoking, hypercholesterolemia, diabetes, which are driving atherosclerosis. But our data would suggest they're not necessarily driving arterial stiffness and the development of hypertension in later life. And we've been interested, therefore, in a couple of novel risk factors for stiffening, and this is one of those, which is deposition of calcium in the arterial wall, which you can see here illustrated on a CT scan. And we showed quite a nice relationship between pulse wave velocity here and the amount of calcium you were getting. And in the accompanying editorial, Verhofjenschik suggested that what we might look at as a paradigm for stiffening of the arteries was disruption of the elastin driven by things like age, heart rate, blood pressure, and perhaps genetic variants, and that may act as a focus for calcification, and that inflammation may be involved in that process of calcification. And it's this role of inflammation in arterial stiffening that I'm going to spend the majority of my session talking about. So it's been accepted for a while that inflammation plays a role in atherosclerosis. If you look at these data from Ridker and there are various other very similar data, what you find is that the people of the highest risk seem to have the highest levels of lipid, but also the highest levels of CRP. And we looked early on in a cohort of individuals collected between Cardiff and Cambridge and found that people with a high CRP seem to have a high pulse wave velocity. And that when you correct it for other confounding factors, CRP seemed to remain predictive of pulse wave velocity. And indeed others have shown similar findings from a range of different populations. This is from the Rotterdam study, slightly older group, but you can see a relationship between CRP and pulse wave velocity. And that remained significant after adjusting the data as well. But the problem with those data is they're cross-sectional. So what you don't know is this just an epiphenomenon, and is the CRP high because of something else and not related to driving stiffness? So we went back to the data we'd looked at in Caerphilly and looked at the relationship between CRP measured 20 years prior to the stiffness measurements and current CRP. And actually we found that both of them seem to be predictive of pulse wave velocity. And if you look at the magnitude of the effect, they were a lot more predictive than things like triglycerides or waist-hip ratio. And this stood out to us as suggesting that maybe inflammation was helping to drive that process of arterial stiffening. Now, one of the questions that's plagued the area of CRP is whether CRP itself is causal in cardiovascular disease, or for that matter arterial stiffening, or is it just a marker of inflammation and things that are going on? And we use what I would call an experiment of nature, or if you want to call it Mendelian randomization, that's fine. But there are various mutations in the CRP encoding genes which are relatively common and which we know cause an alteration in the transcription of the gene, which means that people with certain mutations have, on average, higher CRP levels than those without them. So again, going back to the Caffelli heart study, we looked at some of these published mutations and their relationship to CRP levels and to arterial stiffness. And what we found, first of all, was that the mutations did indeed relate to the levels of CRP. So if you had the mutations, you had higher CRP levels. But actually, those mutations had no relationship whatsoever with arterial stiffness. So what that tells you is that CRP, at least for stiffness, is not causal. It's simply a measure of inflammation and isn't causing damage itself. And indeed, very similar observations have been reported for cardiovascular disease and atherosclerosis as well. So what we then thought about was, well, could you use the paradigm of Koch's postulates, which is most famously applied to bacteria and disease, and could you use those postulates to think about inflammation, arterial stiffness, and therefore work out whether inflammation was causing stiffness or whether it was simply a measure of what was going on in the system? Well, I think you can, and that's what I want to show you, the work that we've done around this area to help understand this. So first of all, you could think about conditions which naturally occur and cause inflammation of blood vessels. Because if inflammation is causing stiffening, then these conditions should be associated with increased stiffness. And you can arbitrarily divide vasculitis or vasculitides into various groups, but most people would divide them into those affecting large and medium-sized arteries and then those affecting the small vessels. So if you look at large vessel vasculitis, and this is a relatively rare condition in the UK, Taschiaceous arteritis, what you find is that patients with vasculitis have increased aortic stiffness compared to controls. And interestingly, they don't have increased stiffness in other vessels like the vessels in the arm. It seems to be a phenomenon predominantly affecting the large blood vessels. Again, rare in the UK, but Kawasaki disease. Patients with Kawasaki disease, and these were children, again, have increased stiffness compared to controlled children. And what was interesting in this study was that stiffness didn't necessarily go away once the disease burnt out and the children no longer had inflammation. Behçet's disease, again, a large vessel vasculitis, and patients with Behçet's disease have quite marked stiffening of the large blood vessels. But the problem with that concept is that these conditions are affecting the arteries directly, whereas inflammation, which we'll all have at the moment, depending on how many colds we've had recently, state of our dentition, things like that, that tends not to affect the arteries directly. So a better paradigm perhaps is to look at conditions not affecting the aorta. And we started off looking at so-called ANCA-associated vasculitis. And what we found was that patients who had active ANCA disease had an increased pulse wave velocity and stiffer blood vessels than controls. And those people who went into remission using a combination of steroids and cyclophosphamide, actually the pulse wave velocity tended to come down and be lower than it was in those people with active disease. But again, you could level the criticism here that even though ANCA affects small blood vessels, there may potentially be some involvement of the large blood vessels as well. So we swapped and looked at rheumatoid arthritis. And we chose this because it's a common disorder, so it's not difficult to get patients, and it's a chronic inflammatory disease, which rarely involves the large arteries in the disease process. It seems to be driven at a synovial level. And what's interesting about rheumatoid arthritis is there have been various suggestions over the years that patients with rheumatoid arthritis actually have an increased risk of cardiovascular disease. And there are various estimates for this, but it appears to be 4 or 5 times that of people without rheumatoid arthritis. And again, if you adjust for other things like cigarette smoking, blood pressure, gender, and age, you find a hazard ratio of about 3 times, suggesting that people with rheumatoid arthritis are about 3-fold more likely to develop myocardial infarction and stroke than people without it. So we looked at a cohort of patients with rheumatoid and found that compared to age and gender matched controls, they had stiffer blood vessels. And again, this predominantly occurred in the aorta rather than the blood vessels in the arm. But that's not very strong evidence of causality. Now, you can't really make people get rheumatoid arthritis. That would be rather unethical. But what you could do is the opposite experiment, and you could take people with rheumatoid arthritis who are being treated for it and study what effect that has on their stiffness. And we're fortunate that we now have very good ways of turning off the disease process in these patients, which when I went to medical school we didn't have. But anti-TNF drugs like etanercept, infliximab, or any of the newer variants are very good at reversing, or not reversing, but preventing further damage in these patients and reversing the inflammatory process. So we followed a cohort of people who had been treated with anti-inflammatory agents and noted that by 4 weeks the pulse wave velocity had effectively normalized and was no different to controls. And since our original observation, a number of other groups have gone on to publish very similar results, suggesting that anti-TNF alpha therapy does indeed reverse pulse wave velocity. This is one of those studies, and probably is the best study, because they looked at people who had rheumatoid as a large cohort and looked at those people who were clinically treated with anti-TNF or for various reasons were unable to get it. So this isn't an RCT, but it's probably the closest we'll come. And they found that the treatment improved the disease. This is a measure of rheumatoid disease, DAS28. It reduced inflammation, and it had a significant effect in reducing pulse wave velocity. So I think the data suggests that anti-inflammatory agents in rheumatoid patients not only improve the disease, but also improve arterial stiffness. And that, again, is very interesting, because if you look at anti-TNF therapy, there's been a suggestion that anti-TNF therapy reduces mortality in patients with rheumatoid. And if you dig into that further and look at cardiovascular mortality, you see about a 30 or 40% reduction in both myocardial infarction and stroke in rheumatoid patients who receive anti-TNF therapy. So there's another way of looking at inflammation in large vessels, and that is to go out and inflame people. And I've suggested that you can't really give them rheumatoid, but you can do something else to them, which we do routinely, which you can vaccinate people. And we know that once you vaccinate people, you get an acute and short-lived inflammatory response in individuals. And this is data from a Greek group, but what you can see here is the effect of Salmonella typhi vaccination in healthy volunteers. And the people given Salmonella typhi vaccine compared to saline got an acute inflammatory response as measured by IL-6 here, but you see a very similar thing with CRP. And what you find is those vaccinated get slightly stiffer blood vessels and compared to those who receive a sham vaccination, suggesting that acutely inflammation can affect the stiffness of your blood vessels. So if inflammation is making your blood vessels stiffer, what's the process driving that inflammation? Well, if you go back to the cartoon of blood vessels, what you'll notice is the blood vessels are lined by a monolayer of cells called the endothelium, and we know that these cells release lots of vasoactive substances. And it's been known for about 40 years that if you change the tone of smooth muscle cells, either poisoning with potassium cyanide or vasoconstricting with noradrenaline, you change the relationship between stress and strain. And what that basically means is you change the stiffness of the blood vessels. And with John Cockcroft and Albert Avolio, we'd published several years ago a study in humans in the iliac artery where we infused monomethyl arginine to block nitric oxide production and found that that made the iliac artery stiffer. So this suggests that nitric oxide coming through endothelial cells may be altering how stiff your blood vessels are. And that's interesting because Arun Ingorani and Patrick Valens and several others had already shown that if you vaccinate people, again using Salmonella typhi, that you impair the ability of endothelial cells to release nitric oxide, assessed here using intraarterial infusions of acetylcholine and GTN. So that led people, including ourselves, to believe that one of the mechanisms by which inflammation might be making your blood vessels stiffer was by impairing the release of nitric oxide from your endothelial cells. And indeed, that's a nice story, and we started accruing evidence that this may well be the case. If you look at endothelial function in rheumatoid patients, which we did, then you find that rheumatoid patients have impaired nitric oxide production compared to control subjects. And if you look at endothelial function in rheumatoid patients who receive anti-TNF therapy, what you find is by 4 weeks, the endothelial function is very similar to controls, and by 12 weeks is not different to controls. And that sort of is a mirror image of what we observed in pulse wave velocity. So it's a very seductive idea that all these changes in stiffness are due to nitric oxide and endothelium. And indeed, this is what many people felt was the case, and thought the chapter on this was closed. But of course, it could be like this. Nothing I've shown you would suggest otherwise. So we decided we'd actually try and work this out and perform an experiment to improve endothelial function and see whether stiffness got better, i.e. not change inflammation, just improve the endothelial dysfunction. And we chose to do this using Tetrahydrobiopterin, or BH4. This is a cofactor for nitric oxide synthase. And there's a lot of evidence that in inflammation, you get uncoupling of NOS, and that NOS starts making free radicals rather than nitric oxide, and that you can recouple that by giving BH4. So we performed both an acute and chronic study in rheumatoid patients looking at the effect of supplementing them with BH4. This was a single dose, and this was over about a week. And these are data that I presented to the Society last year. And what we found was that both acute supplementation and chronic supplementation improved endothelial function as predicted. But you didn't need a degree in statistics to see it had no effect whatsoever on pulse wave velocity. So we had divorced the effect on endothelial function and nitric oxide from the effect on large artery stiffness. And this strongly suggests that we were all wrong, and actually the endothelial dysfunction induced by inflammation is not causal in making the vessels stiffer. It's simply an epiphenomenon. So inflammation is driving both endothelial dysfunction and stiffening of the large arteries. So if it's not endothelial dysfunction, what could be driving it? Well, one of the things that happens when you get inflammation is that your white cells, macrophages, neutrophils, etc., start secreting a number of enzymes like MMP9, MMP2, which can digest elastin. And therefore, it could be that inflammation is making things like MMP9, and that's leading to degradation of elastin in the wall and making the vessel stiffer. I think that's highly unlikely to be responsible for two reasons. First of all, it's inconceivable to me that you get a change in pulse wave velocity within several hours, and it's also inconceivable, given that elastin's got a half-life of about 60 years, that you would restore stiffness to normal within 4 weeks of removing inflammation. So I think this is a non-starter. The other possibility is a direct inflammation of the vessels themselves. And we've been fortunate to collaborate with a group in Norway who've got a large collection of aortic punch biopsies from rheumatoid patients. And when you start looking at these in detail, you find inflammatory cell infiltrates in the wall of the aorta. And therefore we thought, well, if that's what you find at histology, could we look at this in live rheumatoid patients and therefore look at whether they've got inflammation, and perhaps look at what happens when you reduce the amount of inflammation with anti-TNF therapy. And therefore we turn to a technique called PET scanning, or positron emission tomography, which uses labelled glucose, so fluorine labelled glucose, and that causes, that gets taken up into cells, decays, releasing positrons which you can assess. And you can get pretty pictures like this. This is the heart, aorta, and this is an individual with rheumatoid before they were onto anti-TNF alpha therapy, and this is that same individual after they'd completed their course of anti-TNF therapy. And of course you can quantify the amount of inflammation, which is what we've been doing, and these are some preliminary data from an ongoing study. And what we found was basically that the anti-TNF therapy reduces the amount of FDG glucose uptake in the aorta, suggesting the aorta is becoming less inflamed itself. And if you look at the correlation between the change in stiffness and the change in uptake, you find a fairly nice correlation between the two. So it's my own belief that conditions like rheumatoid are leading to stiffer blood vessels because the aorta is involved in the inflammation. It still doesn't answer the mechanistic question about how that leads to a change in the mechanics of the aorta. And my current bet would be it's to do with the amount of ground substance or glycosaminoglycans in the aorta. And here you can see an experiment where an animal's had its aorta inflamed, and this pinkish stuff here is glycosaminoglycan, it's hyaluronic acid. And you can see a lot more in the wall after inflammation. And we know from some very early studies in the 70s that if you alter the amount of glycosaminoglycan in the wall with these various things here, that what happens is you change the stiffness. So having glycosaminoglycan in your aorta is important, and it alters the mechanics. And I think this is what we'll find. I think we'll find it's changes in this that are resulting in changes in hydration status and therefore stiffness of the wall. I just want to finish with a few therapeutic thoughts about how you might apply what we've been doing to try and reduce inflammation and therefore perhaps unstiffen blood vessels. So one of the obvious ways to try and reduce inflammation, particularly in people with cardiovascular disease, is to use statins. And people have suggested that statins have pleiotropic effects and can lower inflammation as assessed by CRP. And indeed, there's a paper published in The Lancet suggesting that atorvastatin was able to improve disease activity, that's clinical disease activity, in patients with rheumatoid arthritis. So we decided we'd have a look at the effect of statins on rheumatoid arthritis. But bearing in mind things I've been taught by John and David, I thought it was appropriate to have a control in this and to actually work out whether this was a true pleiotropic effect or was nothing to do with that and was simply a reduction in cholesterol. So we used this drug, ezetimibe, which is a non-statin cholesterol-lowering drug. And basically what we found is that both simvastatin and ezetimibe reduced pulse wave velocity to a similar degree. And what that suggests to me is it's not anything peculiar about statins, it's just a reduction in cholesterol and perhaps total oxidized LDL, which is what's really driving it. And if you reduce that, you reduce stiffness. And I haven't shown you the data, but you actually reduce inflammation and improve disease activity score in these people as well. And indeed, you can do the same thing with the acute inflammation model, which is what we went on to do. And you find if you vaccinate people, you get an increase in pulse wave velocity. If you vaccinate them in the presence of simvastatin, you get no change at all. And more recently, been working with an experimental compound. This is a compound that GSK owned, P38 MAPK kinase inhibitor called losmapimod. And P38 is one of the kinases which is thought to be important in the downstream signaling pathways of inflammation, particularly in patients with cardiovascular disease. So in collaboration with them, we've been working with this compound, and what we've been able to show is that if you look at endothelial function, then this compound improves endothelial function. This was in patients with hypercholesterolemia, and then we've repeated the experiments we've been doing in rheumatoid and shown that if you assess inflammation in the aorta directly using PET-CT, that you get a reduction in uptake, suggesting an anti-inflammatory effect of this compound. And the nice thing about this compound is it's relatively non-toxic, and therefore it may actually be usable with cardiovascular disease and inflammation, whereas anti-TNF therapy probably isn't going to be really usable outside the paradigm of rheumatoid arthritis and other vasculitis, because it has some downsides to its use. So I'm going to finish there, and I'm just going to make an important point, which is none of what I've done would be possible without the assistance and guidance of others, particularly Kaiser and Carmel, who've done a lot of the work that I presented today, but also the man holding the camera, John Cockcroft, that's why he's not in the photograph, who, as David said, I've worked with for a number of years, and I think successful clinical research in particular is all about collaboration, and it's all about getting a team working together in order to get the experiments done. Thank you very much for your attention.